Hey guys, welcome to your first video lecture for maybe bio this year. Couple things I want to point out about the video that will be important. One, my only way of communicating with you is this mouse. It's the only way I can tell whether you are, or nothing that I can tell, it's the only way that I can point things out to you, so pay attention to the mouse. All right. Secondly, this video is a video, so it's got a pause function. Use that to your advantage, which means if you want to take a break, hit the pause button, come back to the video. If you're taking notes on your PowerPoint, the paper copy, and you want to stop the video so you can write stuff down, stop the video. You can rewind, go back, and watch some part of the video again to make sure that you truly understand it. Alright, so use this video to your advantage. It's going to be very, very helpful. And here we go. Last class, we'd already looked at how water, which is the first part here, was transported in the xylem, and transpiration with the adhesion and cohesion really help it. Today we're going to look at the sugars and the gas exchange as well. Now remember that water is pulled up because of transpiration. As water moves up and leaves the leaf, it pulls up more water molecules along with it. That's because of the cohesive property between the two water molecules. Then we also looked at how water is transported and absorbed from the soil. This happens through osmosis through aquaporins, which are these tiny kind of holes that exist within a root that allow water in from a high concentration to low concentration. We also looked at mineral absorption, which we're going to get into a little bit more detail on the next slide. So mineral absorption is done through our proton pumps as well. The active transport of hydrogen ions out of the cell creates a positive side outside and a negative side on the inside of the cell. This creates a membrane potential and a difference in charge. As a result of this, all the cations outside the cell, which are these positive ions, in this case potassium, they are attracted to the inside of the cell and they enter through a protein without needing energy because they are already attracted to this side. We also use cotransport with the gradient that we've created with the hydrogens. In this scenario, you can see the hydrogens have been pumped outside of the cell through active transport, and they can now bind with a negative ion like this NO3 minus. These both combined can connect with a protein and transport within the cell. So this gradient allows us to do passive transport of cations and cotransport of anions. We also looked at the cell wall and how water enters the root. We've got two routes. We've got the apoplastic route right here, where water enters through the cell wall. And we have the symplastic route, which allows water to enter through the cell itself. And their eventual destination is going to be the xylem, which is going to transport the water to the rest of the plant. I want to talk about the endodermis now. We already discussed this in class. The endodermis is a cell layer that surrounds the xylem, and this actually forces the water into the xylem because it's lined with this impermeable strip. Remember, impermeable means things cannot pass it. This impermeable Caspian strip, which is represented by this purple right here. The water can't go left because the Caspian strip is blocking it. It can't go right because the strip is blocking it, and it can't go backwards because, well, that's where the water's coming from, so it's going to be forced in only one direction, towards the xylem. We already looked at the root anatomy. You can look at these pictures on your own. And we also discussed how the mycorrhizae, the fungi, help the plant and how they are helped back in return by getting nutrients and access to the same water. I do now want to move into transport of sugar in the phloem. We already looked at water in the xylem. Now let's look at the sugar in the phloem. We once again use a proton pump along with cells called plasmodesmata to use cotransport to get the sucrose into the phloem. So in this case, you can see the hydrogen ion is being pumped outside the cell, and outside the cell it is going to combine with a sugar, a sucrose, and then it is going to co-transport it down this protein into the cell. This is how we get sucrose and other sugars into the phloem. Once they're in the phloem, we use mass flow and we use positive pressure. So let's go ahead and talk about what that means. Let's look at the diagram first. Here you can see the leaf, which is the source of the sucrose. That's where it's being made through the process of photosynthesis. And here we've got the root, where we want to store our sucrose. So our ultimate goal is to get the sugar from the leaf down the phloem into the sucrose. So we use something called a source to sink. The source being the leaf, the sink being the root. And the direction of the transport in the phloem is dependent on the plant's needs. Of course, if it needs the sugar to go up the plant, it can do that. But in this case, we're going to look at source to sink. So the active transport of the sucrose into the phloem is what we just discussed, that co-transport with the hydrogen ions. This increases the amount of sugar in this area. Consequently, because there's more sugar, there's less water. This is going back to that hypertonic, hypotonic stuff that we studied earlier in the year. If there's more sugar in this part, where the number 1 is, that means that there is less water. As a result, the water from the xylem is going to be at a higher concentration. This is going to get pumped in through osmosis into the phloem. Once this happens and the water flows in from the xylem cells, this increases the pressure in area number 1. As a result, everything that's up here gets transported down the xylem to the root. The pressure and the amount of water getting pumped in pushes everything down. Once we get down here, all the sucrose ends up in the root and it is stored. We now need to get the water back to the xylem. Well, if you think about it, if we've decreased the amount of sucrose, we now have a high concentration of water. Less solute means less solvent. As a result, the water is just going to osmose right back into the xylem from the phloem. And then it can go up the xylem using the same adhesion and cohesion. And then we have a repeat of the process again and again. So we've looked at this process and studied it in different plants and different animals. All the sap that you see on the side of a tree is just simply the sap is, sorry, lost my train of thought. The sap is simply the food from the phloem that we are sucking out. Maple sugar is developed and processed through the same process, using the same word twice. And that brings us to the end of plant transport. I'm going to be making separate videos for plant nutrition and for hormones in plants, so that way it's a little easier to upload, and secondly, you guys can watch them in smaller bits of information. Thank you for your time, and I hope you enjoyed this different way of receiving your lecture.